Welcome back to our YouTube channel, Histology. In this topic, we will discuss microscopy, electron, and light. Since microscopy gives a view into the tiny world, it has been critical to advancing scientific understanding. Two basic types are involved: light microscopy and electron microscopy, each yielding unique advantages as well as limits. Electron microscopy uses electron beams and electromagnetic lenses, providing much higher resolution than visible light, combined with glass lenses used with light microscopy. Let's take a closer look at the two methods in a little more detail. Optical microscopy. Light microscopy is the most widely used in biology and medicine. It employs visible light to enter a sample with glass lenses for magnification of the image. Its various types are phase contrast, fluorescence, bright field, and dark field microscopy. 1. Bright field microscopy. Probably the simplest of light analysis microscopy, where the illumination is direct. The result is then a bright field background with darker appearing sample. It is very useful for observations and preparation of specimens that are of their own natural color or stained. Typical problems with clear and translucent cells and tissues occur with bright field. 2. Dark field microscopy. It collects only the scattered light, focused at the specimen from the sides. It creates a very contrasting view of the specimen against an optically dark background. When trying to see live, unstained samples, particularly those that are hard to spot in natural light-dark field microscopy, is helpful. Phase contrast microscopy. This technique does not require staining and increases contrast in translucent samples. It gives an excellent image with differently colored hues by taking the differences of refractive index in different parts of the gray material. Because it does not require staining, which might damage the material, phase contrast microscopy is often used in the examination of living cells and tissues. 4. Fluorescence microscopy. This involves labeling particular molecules or structures within a cell with fluorescent dyes or proteins. The excited molecules of these fluorescent proteins emit light at one wavelength when their light of a particular wavelength is excited by another wavelength and will illuminate the labeled structures against the dark background in the cells. Fluorescence microscopy is useful in molecular biology and medical diagnostics, as it provides highly selective observations of particular cellular constituents, such as proteins, DNA, or organelles. Advantages of light microscopy include the ability to be able to observe dynamic processes and living cells. Easy to use and relatively inexpensive. Ability to apply a number of stains and colors to draw attention to particular structural elements. Limitations: the wavelength of light, 200 nanometers, limits the resolution. It cannot see things smaller than that, such as viruses or molecular complexes. Electron microscopy. As light cannot be used to achieve such resolution, the electron microscope employs an electron beam. Electron microscopes are divided into two categories: the transmission electron microscope, TEM, the scanning electron microscope. 1. Transmission electron microscopy, TM. In TM, an electron beam is transmitted through an incredibly thin slice of the specimen. Detecting and utilizing the flow of electrons through the object produces a two-dimensional image with great detail. Since the wavelength of electrons is far shorter than that of light, TEM can get resolutions as low as 0.1 nanometers. Thus, researchers can see single molecules or atoms arranged in a lattice structure of crystals. Some of the inner cell structures that are frequently studied by TEM include the nucleus, mitochondria, and ribosomes. Its extensive application in the study of thin films, nanoparticles, and other nanostructures also falls within the scope of materials science. 2. Scanning electron microscopy. SEM scans the surface of a sample with a focused electron beam. It is not similar to TEM that transfers electrons through the material. The scattering of electrons and other signals, as a result of the interaction of the electrons and the surface atoms, provide a comprehensive three-dimensional view of the surface. In particular, with SM, the surface topography of objects can be studied, such as texture of biological tissues, insects, or other materials like metals and ceramics. Even though the resolution of SEM is much lower than that of TEM, it is still much greater than that of a light microscope. They run from 1 to 20 nanometers. It also allows for bigger samples compared to TEM, since the specimens need not be sliced into ultra-thin slices. The advantages of electron microscopy are: its very high resolution allows it to visualize viruses, single molecules, and entities that exist below the resolution of light microscopy. While TEM gives information about inside structures, SEM gives a complete surface topography. Limitations: live samples can't be studied because the sample has to be fixed, dried, and frequently coated with a layer of metal or some other conducting material. Only samples that can easily pass through the thin sections which the TEM demands can be analyzed. Expensive with some sort of training to use them. A comparison between electron and light microscopy. The only choice between electron and light microscopy is determined by the kind of specimen and scale of observation. Live cells and dynamic processes have been allowed to be examined with the aid of light microscopy. It is much easier to use and much less costly compared to all that, so it has been widely available for routine clinical diagnosis and research. However, its resolution makes challenging observation of finer details in subcellular architecture or tiny objects such as viruses. However, the unmatched resolution of electron microscopy has provided researchers with access to the realm of nanoscale objects. Its ability to expose minute features of cell structures, materials, and even molecular arrangements, it is indispensable, as well as nanotechnology in quite a number of fields, such as structural biology, materials science, and even molecular arrangements. However, it remains less applicable for routine work in biological research, for reasons that include its complexity, high cost, and necessity to use processed or dead materials. Conclusion: our understanding of the microscopic world has been totally revolutionized by both light and electron microscopies. Due to its flexibility and ability to see living cells, light microscopy remains an essential tool in biology. As this is the case, electron microscopy provides resolution necessary to study structures at the molecular level, thus greatly expanding our understanding of the natural world. Both fulfill a specific need in scientific inquiry as they give a contrasting view regarding the composition and properties of materials and living things. Thank you so much for watching us.